thing in, or you have any questions about that? Everybody clear? Clear as mud? Okay, so there's extras up here if you didn't get one, etc. And short of that, I'm out of here. I'll be back mumbling or something. Oop, maybe I should take a few upstairs. Jen, if there are extras, keep them separately because they charge us for blanks, and so then you can turn those back into the secretary if you have blanks, okay? I'll get you guys some upstairs. Okay, here we go upstairs, people. Pass them around. Here you go. And pass them. Here we go. Okay, I'll let you guys sort that out. How you doing, Tanya? Evaluations, so get a form and get it filled out.
Are the forms up front?
Yeah, there should be some around. Hi, evaluations, go get one. Hi, Claire. How you doing? Let's give it a couple more minutes and get it finished up if we can. So thanks for your efforts. There you go. Let's give it one more minute, folks, and then I'll be back in. We'll get started.
Did you get my email about Friday?
Oh, did I not respond?
No.
I'm sorry.
Are you
I remember
free
seeing it.
Friday after 2?
I can't remember, Monica. I don't know my. Hold on. That's why I have an iPhone. I know I'm pretty busy, but I might be able to squeeze you in. If you have questions about stuff, I can generally squeeze you in between something else.
Okay.
So let's see. Hold on. Wednesday, Thursday, Friday. Okay. So
Either like Friday or like Monday's probably. I don't know. Are you?
How
what are you doing next week?
How about 3 o'clock?
Okay. Yeah, that would work.
Are you free?
Mm-hmm.
Okay.
I just have something from 1:30 to 2.
Okay. I'd say maybe I'm meeting with a seminar speaker, but I'd say maybe 3:15. That'll give us 45 minutes. How about that?
Okay.
Okay. Take care. Yep. Okay, here I come. Hide the children. Thank you, Jen. Okay, if you're still filling one out, that's okay. Please give it to Jen when, by the time the class is done, and she'll take it back to the BB office. So thank you all for your comments. I really do appreciate the comments. And as I say, I learn something every term from them. So I really do appreciate them. And, oh, blast it, that's right. We don't have data services here, do we? No, I've got it. I just forgot to pull it up. Okay, so somebody pointed out to me earlier today that Friday says review. That was the original plan, but of course we had class canceled on Wednesday, so Friday is not going to be a review. I'm still going to be doing new material on Friday, okay? I'll also have a nice surprise for you on Friday. I hope that you enjoy it, and I won't say anything more about that than what I just have. So please be here on Friday. What's that? Okay. Whatever it was, a shout from the crowd. Okay, so we're heavily getting into glycogen metabolism. We're going to go through quite a bit of glycogen metabolism today. And one of the things I think and hope that you will see about glycogen metabolism is it's actually very simple. Okay? It's very, very simple. Believe it or not, what you saw on Monday, whatever today is, on Monday was basically breakdown of glycogen. That was basically it. There's a total of 3 enzymes that's involved, and I haven't talked about the third one much, but I'll say a brief word about it today. So to refresh your memory, first of all, we talked about breakdown of glycogen, which involved a reaction called a phosphorylase reaction. That was catalyzed by the enzyme known as glycogen phosphorylase. It used a phosphate to break a bond between 2 adjacent glucoses. It broke an alpha-1,4 bond, and it put a phosphate on position number 1 and made glucose 1-phosphate. Okay? That glucose 1-phosphate, I said, can be converted readily to glucose 6-phosphate, but I didn't tell you how that happens. That's the third enzyme. So if we want to get glucose 1-phosphate into glycolysis, we have to make a glucose 6-phosphate out of it. And that requires action of an enzyme known as phosphoglucomutase. Now that name should probably tell you something about its mechanism of action. What would you suspect is an intermediate in the process of action of that enzyme? Glucose 1,6-bisphosphate, and it is because the name mutase tells us something about how that works. And in fact, glucose 1,6-bisphosphate is released by the enzyme at a low frequency. As far as I know, it doesn't have any significant impact like 2,3-bisphosphoglycerate does, but the same general mechanism and the same phenomenon where we see at a low frequency the escape of the intermediate from the enzyme. Well, in the process of that catalysis, glucose 6-phosphate is produced. That's the primary product of that reaction. And glucose 6-phosphate can be oxidized in glycolysis and then later oxidized further in the citric acid cycle, which we'll talk about next term. Okay? So enzyme 1, glycogen phosphorylase, enzyme 2, and there's no order to these, but obviously you can't have enzyme 2 acting until you have glucose 1-phosphate. Enzyme 2 is phosphoglucomutase. The third enzyme was the one I called debranching enzyme. And debranching enzyme is an important enzyme because glycogen phosphorylase won't cut any closer than about 4 to 5 residues away from a branch. So debranching enzyme is called in. When debranching enzyme comes in, as I said, it has an unusual enzyme. It has 2 activities. One activity is to move 3 glucoses to the end of another branch in an alpha-1,4 linkage. So it's moving from an alpha-1,4 to an alpha-1,4, no change in energy. And it's using water to clip that last glucose off. And the reason it's using water is there's not enough energy in a 1,6 bond to allow the addition of a phosphate. So this is the only place in glycogen metabolism where free glucose is present. Okay? Only place where we get free glucose present, or free glucose released, perhaps I should say. Okay, so that's how we break down glycogen. That's all there is to it. There's the phosphoglucomutase reaction. Glucose 1-phosphate. There's the intermediate, glucose 1,6-bisphosphate. And then the final product, glucose 6-phosphate. All right. Now, we're not going to discuss glycogen phosphorylase structure. We're not going to discuss glycogen phosphorylase mechanism. We are going to talk a little bit about glycogen phosphorylase because glycogen phosphorylase is a very interesting enzyme, not just because of the reaction it catalyzes, but more importantly because of the way that it's regulated. The complexity of glycogen metabolism is not in the breakdown or in the synthesis. You'll see synthesis is almost as simple as breakdown, but rather in how the enzymes that catalyze these reactions are controlled. So the enzyme in breakdown that's controlled is glycogen phosphorylase. And glycogen phosphorylase can exist in two different forms called A and B. A and B. Now, glycogen phosphorylase A is a form that has two phosphates on it. Glycogen phosphorylase B has no phosphates on it. So my first question to you is, how do you suppose the phosphates get on there? What enzyme would be involved? Protein kinase A, and it would be a good guess, and you would be correct. Protein kinase A puts phosphates onto glycogen phosphorylase and converts glycogen phosphorylase from the, actually, I'm actually not telling you the complete truth. Absent-minded professor, right? So protein kinase A plays a role in putting the phosphates on, but not a direct one, okay? So protein kinase A does put a phosphate onto something, but it's not directly onto glycogen phosphorylase. It puts a phosphate onto another enzyme called phosphorylase kinase. The addition of a phosphate to phosphorylase kinase causes phosphorylase kinase to become active. I'll show you a scheme of this in a minute. Okay? And phosphorylase kinase, when it's active, now catalyzes the addition of a phosphate to glycogen phosphorylase to convert the B form into the A form. Addition of a phosphate is all that's necessary to convert the B into the A. Okay? Now, the complexity, and I'll show you the scheme for that in a minute, but the complexity of glycogen phosphorylase is that glycogen phosphorylase can actually exist in 4 different forms. In the A form, it can exist in the R state, and it can exist in the T state. In the B form, it can exist in the R state, and it can exist in the T state. So there's 4 different forms of the enzyme that we can think about. Now I'm going to show you a sort of a simplification of that scheme in just a minute. But we have to think about this. So glycogen phosphorylase A is the form that people typically describe as the, quote, more active form. They describe it as the more active form. That's not completely correct, but you'll see why they say it in a bit. I use that terminology myself, okay? You'll see why it's not completely accurate in just a minute. Okay? Glycogen phosphorylase B is the form that they describe as the less active form. Okay? Now, not surprisingly, what this figure is trying to show you is that addition of phosphates does change the enzyme, and the enzyme can exist in a couple of different states. Okay? This figure now shows the 4 different forms of glycogen phosphorylase. Glycogen phosphorylase, as I said, if we look on the left side, we have the B form, which has no phosphates. If we look on the right, we see the A form, which has 2 phosphates. You see this is a dimer of 2 identical subunits, so each subunit gets 1 phosphate to become in the A form. Now, this figure also shows you that both the A and the B form can exist in an R and a T state. Now, what we know about R and T states, which state would you suppose is the more active, the R state or the T state? The R state, okay? So for both the A and the B forms of glycogen phosphorylase, the R form of that will be the more active form. For glycogen phosphorylase in the T state, okay, that will be the less active form, again, for both the A and the B forms. So the real key to understanding this is, well, how do these forms interchange? Now, there's a very important point that you probably haven't picked up on, but I'm going to make it for you here. We've talked in class a little bit about how enzymes can be modified. They can be modified by covalent modification. They can also change their shape by allosteric interaction with small molecules. Okay? Now, what I didn't hammer home to you before, but I will hammer home to you now, is the only thing that converts an enzyme from the R state to the T state is allosteric interactions. When we think about allosteric interactions, we're talking about conversions between R and T states. Well, you sort of saw that with ATCase. We added ATP, you got it in the R state. You added CTP, you got it in the T state. All right? What I didn't tell you was covalent modification does not, underline not, change R and T. So when we see R and T state, as we do on this figure, going up and down, the up and down changes between R and T for either form have to occur as a result of allosteric interactions, or, as we will see, the built-in structure of the enzyme. You'll see what that means in a minute. If we want to go from B to A or A to B, we've either got to put a phosphate on, going to the right, or take a phosphate off, going to the left. So if I wanted to go from this form to this form, I would have to either go right and then down, or down and then right. Either is possible. But I couldn't go diagonally. Couldn't do them both at once. Okay. Is that clear? Now, this stupid microphone, I'm sorry. All right. What's going to happen then is we now have to consider, you've seen first of all how the covalent modifications make A and B. All right? Or B to A, etc. All right? Now we need to talk about how this up and down business occurs. And this is where we're going to get the clue about why we think about A form being more active and the B form being less active. Your book makes a point of talking about phosphorylase B in muscle versus liver, and to be honest, we're not going to differentiate tissues. But these happen to be the ones that were studied, and there's reasons why that we won't go into here. Okay? So let's look at glycogen phosphorylase B. We're focusing at the moment only on glycogen phosphorylase B. This is the form that does not have the phosphate. And this form, when we look at this, we now see the allosteric modifiers of this enzyme. If we want to convert it into the R state, it takes AMP. Now we saw AMP affecting glycolysis. What was AMP an indicator of? Low energy, right? This makes sense. Low energy should convert this enzyme into a more active form, because what this enzyme is going to do is it's going to break down glycogen and give us things that are going to feed into glycolysis. Low energy says we need more energy. We break down glycogen, we get glucose, we have more energy. Okay? This guy is turned off allosterically by two different compounds. One, ATP. What was ATP a measure of? High energy, right? Okay? So high energy should, in fact, turn this enzyme off. We don't want to be breaking down our glycogen when we have plenty of energy. This works out very nicely. Glucose 6-phosphate turns this enzyme off. Now what we haven't said, but I'll tell you here, is that when glycolysis gets plugged up, we start seeing the intermediates accumulating, and one of those is glucose 6-phosphate. So glucose 6-phosphate is giving us an indication that glycolysis is plugged up, and it's probably plugged up because there's plenty of energy. Again, consistent with the energy idea. Make sense? Now, the key, the reason people think about the B form being less active is usually there's very little AMP in cells, and usually there's much more of these two, ATP and glucose 6-phosphate, meaning that usually this enzyme, this form of the enzyme, is found in the T state. And the T state is the much less active form of the enzyme. So it's not inaccurate to say the B form is really the less active form, but it is inaccurate to say that it can't be active. It can. Now, if I have time, I'll give you an example about how that might be important to consider later, especially in muscle. Okay, questions about this? Okay. Let's look at another form, that is the phosphorylase, glycogen phosphorylase A. And by the way, I call them glycogen phosphorylase A. Most books just call them phosphorylase A or phosphorylase B. That's all they call them. Okay? So you'll see your book will typically refer to it as phosphorylase A and B. I prefer to put glycogen phosphorylase A and glycogen phosphorylase B in that. You can abbreviate those as GPa and GPb if you wish. That's fine by me. Okay. Now, this form is interesting. Look at the allosteric modifiers. If we want to convert glycogen phosphorylase A from the T state into the R state, okay, nothing is required. Meaning that this enzyme, as it's sitting there, is already built to be in the R state. Well, how do I convert R to T? Well, you can see it here if glucose is present. Now, when glucose is present, what does that mean about the cell's energy state? Again, it's very high. We've got plenty of glucose. The cell is at a high energy state, and this enzyme gets turned off. Okay? Well, it turns out for this enzyme, cells don't usually have much glucose sitting around. And as a consequence, most of the time the enzyme, as long as there's no glucose, is sitting there in the R state. Only when glucose is present is it converted to the T state. Now this turns out to be a very important thing for a muscle, for a liver cell. If you think about it, the liver was one of two organs of the body that makes glucose, the other being the kidney, a part of the kidney. The liver is the main organ that makes glucose. Your muscles don't make it, your skin doesn't make it, your hair cells don't make it. Only your liver and the cortex of your kidney make it. Okay? Now it's very important then, we think about the liver is making all this glucose, the body is I'm out running, I'm jogging, I'm doing my thing, and I'm burning glucose, right? So my liver gets that signal and says, hey, he's running, we had better start dumping out glucose. So my liver starts breaking down glycogen and starts synthesizing glucose, both at the same time, so it can dump them out into my bloodstream. Make sense? Okay? Well, I stop running. Okay? So I stop, and when I stop, my muscles start catching up. They stop taking all this glucose in, and what happens to my blood glucose levels? They start to rise, right? As my blood glucose levels rise, my liver has a harder time pushing more glucose out because, hey, there's already plenty of glucose out there. So what happens to the concentration of glucose in my liver cell? It goes up, and this is a perfect signal to tell the cell, quit breaking down your glycogen. He's stopped running. Okay? So these controls that are built into these enzymes are really interesting and really neat. You can understand something about how your body works and what your body's doing when it's supplying or not supplying glucose as appropriate. Now what we will see actually next term will be that how all of these things will tie into other metabolic pathways. So much as you might like to think we won't have to think about this again, actually you will get to think about it again next term. Oh joy, oh boy, right? Okay. All right. So that's what's happening with those. Are there any questions on that before I move forward? Okay. We're getting there. All right. So that's what's happening with the regulation of glycogen phosphorylase. So we think of the A form as being more active because cells don't typically have much glucose there. Well, how does a cell decide which one is more active, which one to have? And the answer is the cell doesn't completely decide that on its own. The cell decides it based partly on allosteric modifiers, but what's the other factor? What's the difference between the A form and the B form? Phosphorylation. What controls phosphorylation? Hormones, hormones, hormones. Okay? So you've seen how epinephrine affects this. Okay? It turns out epinephrine works on your liver very nicely. Your muscles don't respond to epinephrine. I didn't mention that to you, but I'll tell you here. Your muscles don't respond to epinephrine. That's kind of weird. Okay? They don't respond to epinephrine. But they do respond to something called glucagon. Glucagon is another hormone that affects muscle cells and liver cells. Okay? Glucagon has exactly the same effect. It turns on the phosphorylation of glycogen phosphorylase B to make glycogen phosphorylase A, and it does it by activating protein kinase A, and I'll show you the scheme again for that in a second. Okay, so that's what's happening. Now let's look at this overall scheme that's here, okay? All right? Oh, let me show you the scheme, and I'll come back to that. Okay, so here's the scheme. This elaborates now a little bit further the scheme you saw earlier of epinephrine signaling. You can also think of the same thing as happening with glucagon. Okay? And I said those two backwards, didn't I? Okay, sorry. Glucagon's present in, it affects liver. Epinephrine affects muscle. I said it backwards. Glucagon does not affect muscle. Sorry. Yeah. Absent-minded professor. Not a good day. Okay. Yeah, so glucagon does not affect muscle. I said it was epinephrine. It's actually glucagon that does not affect muscle. It's epinephrine that affects muscle. Okay. I could have told you the book is wrong. Yeah, I'm the great authority. No, I'm wrong, and I've been known to be wrong. And I have no problem saying I'm wrong. The slide is correct. Yeah. Yes, ma'am. Glucagon does not affect both of them. Epinephrine affects both of them. Epinephrine can affect, so I said it backwards. I said it was epinephrine that affected only liver, but it's epinephrine that affects both, and glucagon that affects only liver. Now, glucagon actually is an interesting hormone. I'll just say a brief word about it. Glucagon is a peptide hormone, meaning it's a polypeptide, and it's synthesized not in response to fright. So you get chased by the grizzly bear, and your epinephrine is what's going to be produced because you need that quickly and you need that now. Glucagon is not synthesized, is not produced under those conditions. Glucagon is produced when your body is running low of glucose. When you're getting hungry and your blood glucose levels fall, glucagon is being released. And it's being released so that the liver will start pumping out some glucose to raise those blood glucose levels. Okay? I like to think of glucagon as meaning glucose all gone. It's a very easy way to remember. When blood glucose levels fall, glucagon is being produced. Okay, well, either glucagon or epinephrine, they both have the same effect if we're talking about the appropriate cell. In this case, let's say we're talking about liver. In the liver, we see exactly this thing happening. That is, we have the signaling, we have the activation of the G protein, the activation of adenylate cyclase. You've seen this before. Production of cyclic AMP, activation of protein kinase A, and here's that intermediate enzyme that I described before that's essential for understanding the conversion of phosphorylase B to phosphorylase A. This enzyme is called glycogen phosphorylase kinase, or as your book calls it, phosphorylase kinase. Same thing. This enzyme is present without a phosphate, it's inactive. Present with a phosphate, it's active. So phosphorylation of phosphorylase kinase causes it to become active. And since it's a kinase, when it's a kinase, it turns around and phosphorylates glycogen phosphorylase B to make glycogen phosphorylase A. Notice that protein kinase A does not directly do that. Protein kinase A does many things, but it does not directly phosphorylate glycogen phosphorylase B to A. Okay? So now we start to see that picture expanding a little bit of that signaling pathway that we saw before, and we'll see it expand again when we talk about synthesis in a few minutes. Questions about this? Yes? Will it try to phosphorylate all of the phosphorylase B? So as long as it's active, it potentially can, yes. So the relative amounts of B and A are one of the control factors for what the cell is trying to do. So if you're really hungry or you're really being scared, then you're going to convert a lot of this into a lot of this. Okay? And it actually happens fairly quickly. Okay, now the thing I skipped over I want to come back and look at, and that is phosphorylase kinase, or glycogen phosphorylase kinase. This looks really complicated, and it's actually really simple. First of all, it's just telling you that it catalyzes the conversion of phosphorylase B into phosphorylase A. That's what the reaction you see on the right. All right? All the business on the left is telling us that there's 2 steps to the full activation of this enzyme. 2 steps. Okay? One step is phosphorylation. That's the part you've already seen. Protein kinase A can put phosphates onto it, and that makes it what this figure says, partly active. The other step in the process is calcium binding. When calcium and phosphorylation happen, the enzyme is fully active and ready to go out there and do business converting B to A. If I have only phosphorylation happening, it's partly active, meaning not very active. If I have only calcium here, it's only partly active, not fully active. So it's a dumb figure. It's trying to communicate that to you, and in the process, it ends up confusing people. Now, there's more than just a trivial note here. There's something interesting about muscles. Okay? Muscles. And it relates to that flight or fright phenomenon that we talked about before. Let's imagine I am standing here, and all of a sudden, someone in the front row scares the crap out of me. Okay? And I decide I got to get away from this person, and I'm going to go running. Okay? My first instinct is I've got to get out of here. I've got to get out of here as fast as I possibly can. All right? My muscles, what's going to happen? My muscles start contracting. I'm running. I'm getting away from here, right? What do my muscles need for energy? ATP. And where do they get their ATP? Breakdown of glucose. Okay? That means they need a lot of glucose. That's number one. Well, when I take off on a mad dash, I can be burning my glucose in my muscle cells, okay, faster than my epinephrine can even get the signal to my liver to start dumping stuff. That takes a few seconds. Meanwhile, I'm scared of whatever's going on. I want to get as far away as fast as I can. This would happen if I'm running a 100-yard dash, for example. Same phenomenon. Okay? So I need glucose almost instantly. Cells don't keep a lot of glucose floating around because glucose is a poison. Well, what's their stores of glucose? Muscles have glycogen. Okay? What signals muscular contraction? We talked about it in here before. What molecule signals muscular contraction? Calcium. So, when my nervous system says contract, the muscle cells release calcium. The very first thing that happens, in addition to causing the muscles to contract, is that extra calcium is binding to phosphorylase kinase and making it active almost instantly, at least partly active. Meanwhile, we start waiting for our bloodstream to bring the epinephrine to get fully active. But even before it's fully active, it's partly active right here. I start breaking down glycogen the second I start contracting muscle. In other words, I am giving glucose immediately as I need it. Now, it's not the most efficient way, but shortly that'll happen because once my epinephrine hits those cells, in fact, it's going to cause that additional contraction. I'm sorry, that additional activation, which will produce more glucose, so I get more contraction. Yes, sir?
Does liver phosphorylase kinase also require calcium to be fully active?
Does liver phosphorylase kinase also require calcium? As far as I know, it does, yes. So it's the same in both.
Would
Yeah.
it be fair to say then, because I can't think of why calcium would be high in your liver cells particularly, is it?
Yeah. I don't have an answer to the question, but as far as I know, but they both require it. Yeah. Okay. Does that make sense? Yes. Yeah. Yeah.
So on this little diagram here, it looks like it's going from the T state of B to the R state of A.
It's not intended to show you that. No.
It's supposed to
No.
go from
B
B to A.
to A.
Okay,
Yep.
so the calcium doesn't like switch it from T to R.
That's right. That's right. So the only thing that switches T to R is allosterism. Phosphorylation cannot do that. Okay. So that is, in summary, in a nutshell, what's happening in the breakdown of glycogen. Let's turn our attention now, we've got about 10 minutes, to talk about the synthesis of glycogen. Okay. Now, the synthesis of glycogen, in a lot of ways, is very simple as well. But it's not the exact reverse of glycogen breakdown. There's a very good reason why. If you think about it, glycogen breakdown, we were producing this energy, right? And we said that making that bond had to put some energy into the molecule, right? We had that 1,4 bond that had that energy. Somehow we've got to put that energy into that molecule. So cells have a way of doing that, and they use an intermediate called UDP glucose. I showed you this earlier in the term, and I said this was an activated intermediate. And I'll remind you that an activated intermediate is a molecule that has a high energy bond. And it uses the energy of that bond to donate part of itself to something else. So an activated intermediate is a molecule that has a high energy bond. It uses the energy of that bond to donate a part of itself to something else. And the part of itself here that's being donated is the glucose. This is what we use to add glucose to a growing glycogen chain. Well, where does UDP glucose come from? That means for synthesis, we've got one more enzyme we've got to consider, the enzyme that helps us to make UDP glucose. That enzyme has a mouthful of a name. It's called UDP glucose pyrophosphorylase. Blah, what a name. And that is shown right here. Okay? UDP glucose pyrophosphorylase is an enzyme that probably we could have another name for. Do I have any suggestions? A lot more suggestions than before. We had a male name. I think we should have a female name here. Cindy. Cindy. I like Cindy. Okay. Second that. Is your name Cindy? Oh, okay. Do we have any Cindys in here? There's no Cindys in here? Then it's a perfect name. Okay. All right. This enzyme henceforth shall be known as Cindy. Okay? Now, Cindy catalyzes the joining of UDP from the UTP that we start with. Okay? Oh, I'm sorry, I've got the wrong reaction. Blah. I should be paying attention to what I'm doing here. Here's the reaction. Sorry. This is really not my day. End of the term is near. The reaction catalyzed by Cindy is here. All right? Last slide was wrong. Sorry. Cindy catalyzes the addition of a glucose 1-phosphate. You've seen that before. If I needed to make glucose 1-phosphate, how would I make it? What would I start with? I haven't heard anybody say the magic word. And it's not please. That's what your mother always says. Glucose 6-phosphate. Well, how would I make glucose 1-phosphate from glucose 6-phosphate? Phosphoglucomutase, right? So that enzyme that I can use to dump things into glycolysis can also be used to run the reverse reaction to make glucose 1-phosphate. That's one way I can make it. Okay? All right. Glucose 1-phosphate plus UTP gives me UDP glucose plus what's called pyrophosphate, PP. 2 phosphates joined together. If you add up the phosphates here, there's 1 phosphate here, there's 3 here, there's 2 here, and there's 2 here. The phosphates add up. Okay, this intermediate then is used to add glucose to a growing glycogen chain. And that was what was in the slide I was showing you. That is this guy right here. Okay? This reaction, which I incorrectly said was catalyzed by Cindy, is actually catalyzed by an enzyme with a very simple name. It's called Glycogen Synthase. Synthase. Glycogen synthase starts with UDP glucose and a glycogen chain. It adds the glucose in a 1,4 configuration. And the reason it's able to do that was because UDP glucose was an activated intermediate that had a lot of energy in that bond right there. I'm sorry, in that bond right there. And that allowed the formation of that high energy alpha 1,4 linkage. That's how that bond got made. We're almost there. We've synthesized glycogen. And because we've synthesized glycogen, we're not quite done because why? We haven't talked about branches. And branches have a mouthful of a name as well. And, oh, come on, where's it at? That was not what I wanted. Don't have it on here. Okay. Well, I'm sorry, I thought I had a figure on there. So, branching enzyme catalyzes the formation of branching. So it grabs a bundle, and your book goes into all kinds of detail, and people they pee their pants over how many glucoses from the end it grabs and where it puts it and how it attaches it. Basically, it grabs a bundle of glucoses from the end that's in a 1,4, and it transfers it over here and makes a single 1,6 bond that's the branch. Okay? That's all it is. So branching enzyme does that. So as a result of having branching enzyme, we now have glycogen. So the synthesis of glycogen required 4 things. Phosphoglucomutase to give me glucose 1-phosphate. Cindy, glycogen synthase, and branching enzyme. With those 4 enzymes, I can make glycogen. Okay. Now, as you might imagine, it can't be just that simple. It's biochemistry. So we have to understand something about how the synthesis is regulated as well. Now, this makes sense. We regulate the breakdown. We need to regulate the synthesis. We saw the same thing happening with glycolysis and gluconeogenesis. Why did we want to have those two regulated the way they were? What were we trying to prevent? A futile cycle, right? We don't want to have them both going on at once. So what kind of regulation did we have? We gave it a name. Reciprocal regulation, right? What did reciprocal regulation mean? Single thing has opposite effects on the two pathways, right? What you're going to see in glycogen metabolism is that there's a lot of things that are paralleled in terms of having opposite effects. Okay? They come down to phosphorylation. That's the key. Phosphorylation is the key. So, here's the scheme you saw before. Here we go down and we make glycogen phosphorylase B into glycogen phosphorylase A, and we break it down. Now we've superimposed on here the regulation of glycogen synthesis. It turns out glycogen synthase is the enzyme that is regulated, and it exists in two forms as well, with a phosphate and without a phosphate. Now, here's where you'll get a little confused. Books use A, okay, to designate the more active form of an enzyme. So we saw glycogen phosphorylase A was what we described as the more active form of the enzyme. Glycogen synthase A is the more active form of the enzyme. However, glycogen synthase A has no phosphates on it, whereas glycogen phosphorylase A has phosphates on it. So A doesn't indicate phosphates. It indicates more active versus less active. Addition of phosphates to glycogen synthase occurs by protein kinase A, there's our friend, and it turns the enzyme down or off. Opposite effects. Addition of phosphates to glycogen phosphorylase made it more active. Addition of phosphates to glycogen synthase makes it less active. One signal, opposite effects. I turn off glycogen breakdown when I turn on glycogen synthesis. I turn off glycogen synthesis when I turn on glycogen breakdown. They're opposite. Yes, ma'am?
You're saying less active, and then up there it says inactive. So
Technically, all these are less active, but again, it's just sloppy nomenclature. Yeah. Okay, we're not quite there. Oh, yeah, I guess we are. Any questions about that? Yeah, quick question. Yeah. C-I-N-D-Y. How about that? Cindy. We'll call it Cindy. All right, guys. Take care. Let's see. How do you want this? Get out of the Mac.
Is it on Windows? Get out of the Mac.
Get out of the Mac. Okay.
What the hell is that?
That should do it.
Do it.
How you doing?
I'm all right. How you doing?
Good. The end is near.
Cindy. C-I-N-D-Y.
That's right.
Got it?
Yeah.
Hi.
So it's grading.
That needs to go in the main office. I'll lose that. Yeah.
Okay.
Yeah. Oh, here you go. And get that out of your way, and we're off and running. Hi.
I'll be gone on Friday. Will I miss anything that I won't get from the video?
I hope not.
Okay. Kevin,
Yes.
looking when I get a note card?
Okay. Did you miss class the other day? Yeah. You'll have to get it from me later. Yeah.
I have a question.
Okay.
Where do we return, or where do we?
Those were due at the beginning of class today.
Okay.
Yeah. So if you want to sweet talk the secretary over in the main office, she may take it. You'll talk to
I thought
her.
we were supposed to bring them to class.
No, I never want them in class because I'll lose them. Okay?
Should we go to your office to get our regraded?
No, you'll get them out of the main office when they're ready.
Okay.
Okay?
I think my mom got it at the employee store.
How you doing, Lauren?
They say no. Good.
Good. If I could turn this off, I'm not sure what's happening. Turn off.
I'm bringing you your letter today.
Good. Good.
I was going to print that right now. If you're going to be at the library, I live there. So
Hi.
To ask you about this, we go to your office hours, is that right? Is that correct?